Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss the historical background of plant tissue culture technique. So what are the major discoveries in this field? At first, conceptual foundations and early experiments from 1902 to 1930s. At first, in 1902, German botanist Gottlieb Haberland, who is widely regarded as the father of plant tissue culture because he laid the foundation of this field, and he successfully elaborated the concept of this cellular totipotency. What is cellular totipotency? Totipotency is the inherent capacity or capability of plant cell to grow into a complete new individual plant. We can grow a complete individual plant from a single tissue, meristematic tissue of any plant, which is known as explant. From plant we can collect explant from shoot, root tip or leaf. We can cultivate that explant using synthetic medium and in vitro condition, and we can cultivate and grow a new plant individual. Okay. In 1904, Hanning pioneered the concept of in vitro embryo culture. In vitro means inside the lab environment. We can culture embryo by successfully raising isolated embryos into complete plant. Then in 1922, scientists W.J. Robbins and W. Cort independently achieved the continuous cultivation of isolated root tips. So these are some early experiments on this field. Next, breakthrough era from 1930 to 40s. At first, Philip R. White successfully developed synthetic nutrient medium for culturing isolated tomato roots. So he discovered the composition of nutrient medium for growing our tissues, for culturing our tissue. Around the same time, Gottfried and Knopfcoat successfully cultivated plant tissue for unlimited period using natural auxin and vitamins. So this auxin and vitamins are required for tissue culture. He discovered these two chemicals are required for culturing tissues. In 1941, J. van Overbeek discovered that adding coconut milk to culture medium drastically stimulated the growth and development of plant embryos, leading to later discoveries of plant hormones, vital plant hormones. So he discovered that the coconut milk can be a vital component of tissue culture medium. Next, in 1955, Skoog and Miller isolated kinetin, which is a cytokinin. They discovered the principle of hormonal control, providing how the ratio of cytokinin to auxin detects whether a callus forms root or shoot. So Skoog and Miller discovered that the cytokinin auxin ratio plays vital role in root or shoot development. Okay. In 1962, Murasis and Skoog developed widely used MS medium or Murasis-Skoog medium. This highly nutritious, chemically defined medium unlocked the ability to culture a massive variety of species which are previously resistant to in vitro growth. Okay. Next, advanced applications. In 1960s, G. Morrell revolutionized horticulture by successfully utilizing root tip or meristem culture to regenerate virus-free Cymbidium orchids, sparking modern micropropagation. So he used meristem culture method to generate or to produce virus-free plants of orchids. In 1964, S. Guha and Maheshwari discovered anther culture technique or haploid culture technique. From 1970 to 80s, protoplast culture and somatic hybridization allowed scientists to remove plant cell wall and fuse cell from different species to create novel hybrids. Okay. Now genetic engineering and this modern era in 1980s, the understanding that Agrobacterium tumefaciens naturally integrates the plant genome, integrates DNA into plant genome, led to the development of first transgenic plant. And in present day, the tissue culture has diversified into complex automated system like bioreactor micropropagation, cryopreservation for germplasm conservation, genome editing using CRISPR-Cas9 technique, all of which rely directly on foundational principles of totipotency, which was established decades ago. So we should know these discoveries and foundation and about the past experiments before reading the plant tissue culture method. So I hope this video provide some benefits to you, and this video is very important to know the introduction part of plant tissue culture. And with this I am concluding my lecture video here. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.